Today we will explain the effects of heavy metal ions on BY-2 cell mitosis. Effects of heavy metals, cadmium, lead, arsenic, and mercury, were investigated on specific stages of the cell cycle of BY-2 cells. BY-2 cells were treated with these heavy metals at specific concentration, and the resulting cells were fixed, stained, and imaged. The relative cell cycle lengths and abnormalities during specific stages of mitosis of these cells were compared with those grown in metal ion-free solution. Cadmium was found to cause chromosomal aberrations and subsequent cell cycle arrest in prophase, lead in metaphase, arsenic in anaphase, and mercury in telophase. Due to the specificity of heavy metal ions to stages of the cell cycle in BY-2 cells, these results can be extended to the development of treatments for cancers in animal tissue. Our background information included heavy metals can be high in soil due to geology and industrial waste. High concentration of these metals has been known to cause stress and cell damage in plant and animal organisms. Cadmium inhibits plant root and shoot root growth, manifesting as a result of chromosomal aberration, which then correlates with abnormalities in prophase, metaphase, and anaphase.
Here on this slide, we show the cell cycle of a healthy animal cell. Prophase is marked by DNA in the form of chromosomes, and the nuclear envelope is still intact at this point. During the next phase, metaphase, all of the chromosomes line up along the equatorial plane. When all of the naked kinetochores on the chromosomes have been attached to microtubules, the cell enters anaphase, during which the chromatids are separated by the microtubules. During the last phase, telophase, the sister chromatids reach the poles of the cell, and the nuclear membrane begins to reassemble to begin the separation of the cell into two daughter cells.
Our research question that we developed for this experiment is, how does the presence of heavy metals ions other than cadmium affect the growth of BY-2 cells? From this research question, we developed a hypothesis stating high concentrations of different heavy metals will disrupt some proteins involved in mitosis more than others. We also proposed that heavy metal treatments will interfere with specific stages of mitosis, leading to an accumulation of cells in prophase, metaphase, anaphase, or telophase.
In order to test their effects on mitosis, heavy metals were used to treat BY-2 cells in specific concentrations as specified in this slide. This involved one positive control, 3 experimental groups, and 1 negative control group. Since cadmium ions had been shown by prior literature to affect the growth of plant cells in specific stages of the cell cycle, BY-2 cells treated with culture media with cadmium was a positive control. BY-2 cells were also put into groups that were treated with culture media with lead ions only, arsenic ions only, and mercury ions only. These served as our experimental groups. Since we anticipate that BY-2 cells should grow normally in the absence of metal ions, BY-2 cells treated with only culture media was our negative control. All of the groups were left in culture media for 1 day and were stained as specified in the slide. Then the various groups were analyzed and compared by taking a sample size n equals 50 of each. In the figure to the left, we show the relative lengths of the individual stages of the normal BY-2 cell cycle. In the figure to the right, we show the relative lengths of each of the cell cycle stages for each of the non-control groups. Though no one heavy metal treatment caused BY-2 cells to have significantly higher time spent in interphase than the others, cadmium caused cells to be observed more frequently in prophase, lead in metaphase, arsenic in anaphase, and mercury in telophase. In addition, whereas the number of mitotic abnormalities in each of the mitotic stages in the negative control were, as expected, nearly 0, aberrations in prophase increased with the treatment of cadmium, those in metaphase increased with lead treatment, those in anaphase increased with arsenic treatment, and those in telophase increased with mercury treatment.
The data shown in our graphs suggests that the treatment of BY-2 cells with heavy metals had the following effects. Cadmium caused an increase in abnormalities in prophase, lead caused an increase in abnormalities in metaphase, arsenic caused an increase in abnormalities in anaphase, mercury caused an increase in abnormalities in telophase. Treated BY-2 cells arrest in different portions of the cell cycle due to chromosomal abnormalities affecting specific molecules involved in each mitotic phase. These images found from the cadmium research paper represent the 4 abnormalities that occurred in metaphase and anaphase after their cadmium treatment. Like our results with BY-2 cells, this heavy metal treatment directly affected the cell cycle process. These abnormalities include multivalence in prophase in picture A, chromosome stitching occurred at anaphase in picture B, bridging between microtubules at anaphase in picture C, and laggard at anaphase in picture D.
So what are the implications of these results? Well, the first one would be that plants can be an effective method of screening potential drugs for the use in animals. In addition, since heavy metals affect specific stages of the BY-2 cell cycle, they can be used to develop treatments to target animal cells as well. Data on the effects of heavy metals on animal cells can then be used to develop cancer treatments to directly target specific points of the cell cycle during the growth of a tumor. However, this experiment still has some limitations. For instance, since live imaging was not performed, the time spent in each phase and the total time during the cell cycle was not determined. In addition, an experiment on only BY-2 cells does not guarantee insight into other types of plant or animal cells. The following is a list of the references used throughout this presentation. We hope this presentation was informative for you, and thank you for your time.